Did you know that our reliance on animals for pharmaceutical testing may be over sooner than you'd expect? Animal testing has been an important practice this past few decades when manufacturing medicine that is safe for human use, and one that has become more and more controversial as the public becomes more conscious of the consequences it has on the animals utilized for this type of research. However, while we may not yet be rid of the necessity of it, promising alternatives have begun to arise in recent years. Which brings us to one of the most promising ones, OOCs. First successfully produced in 2010 by Dr. Donald Ingeber, OOCs, an abbreviation that stands for Organ on a Chip, is a technology still in its infancy that allows us to create a multi-channel 3D microfluidic cell culture integrated circuit. Or, in simpler terms, it is a chip that simulates the physiology of an organ, as well as its mechanics and responses to stimuli. A small-scale artificial organ capable to be used for in vivo testing, that is, the testing on living organisms. With this new technology, we can test the effect a drug could have on a human organ without putting anyone at risk. And results from the first few attempts at using this technology have been very promising on how accurate it is. This is an extremely important development for drug manufacturing, as we have seen multiple times how testing on model organisms, such as rats and mice, among others, falls short, and the passing of drugs with positive results in this stage have resulted in severe consequences, even deaths once they arrive on the human testing phase. Or cases in which life-saving drugs would not have passed if the current laws requiring an animal testing phase had been in place when they were discovered, such as tamoxifen and imatinib, effective cancer treatments that had immense adverse effects when tested on multiple animal-human models, but proved beneficial to humans. Other examples that might not exist today if the current laws dictating drug testing existed are aspirin and penicillin, as well as cyclosporin, a drug utilized to treat autoimmune disorders and prevent the rejection of transplants. Unfortunately, our reliance on animals is not yet over, as they provide a way for us to research the impact a drug may have across generations. And that's without mentioning the fact that we have not been able to create an OOC that shows the impact across the body as a whole, as our bodies are made of deeply interconnected systems. And there hasn't been enough research to establish a standard in the properties of these chips. However, I for one am very excited to see where this technology leads us,